We are on the subject of basal lamina. We have seen the ultrastructure of basal lamina, what molecules make this substance or the substrate. Now let's talk about some additional functions of basal lamina. One of them being basal lamina acting as a barrier for cellular movement. It needs to restrict cells. There are, of course, some exceptions. We'll talk about them. Additionally, basal lamina plays an important role in tissue regeneration. For example, when cells die, if the basal lamina persists, it has a memory and it can transfer that information to cells, allowing them to perform their normal functions. So let's look at that. Basal lamina, for example, I've just said as it forms a function of a barrier. It will prevent cells from crossing it. Of course, there are certain exceptions to the rule. We know that our immune cells, they have to get to the site of infection or where there has been a damage. In order to do that, they need to cross basal lamina. We will see later on that these cells actually have special proteases present on their membrane that allow them to cut basal lamina at different sites. But that would be in a different module. So also nerve cells can also transverse basal lamina if they are required to do that. Generally, it is restricting basal lamina is restricting fibroblasts from entering a different tissue. Fibroblasts are very proliferative cells, and if they invade a tissue, they can cause fibrosis and also prevent that organ or tissue from performing its normal function. So the additional function that I really want to talk about basal lamina in context of basal lamina is tissue regeneration. When tissue, for example, muscles or nerves and epithelia are damaged, the basal lamina survives and provides a scaffold along which regenerating cells can migrate. So basically, the cells have been lost. Now cells, in order to restore the function of damaged tissue, new cells have to move in. And how would these cells move in? What would tell them where to go? That function is performed by basal lamina. It also results in the original tissue architecture to be reconstructed readily. In skin or cornea, the basal lamina becomes chemically altered after injury. For example, addition of fibronectin, which promotes cell migration, which is essential for wound healing purposes. Here, let's look at another example. We have muscle cell, which is innervated by nerve, forming a neuromuscular junction. Basal lamina plays an instructive role in regeneration of neuromuscular junctions, the site where the nerve terminals of a motor neuron make a chemical synapses with the skeletal muscles, which is this region. The basal lamina separates the nerve and muscle cell, and the plasma membranes at the synapses and the synaptic region of lamina has distinctive chemical character or nature. For example, at the synaptic site, there is a special isoform of collagen and also laminin, special isoform of laminin molecule, another proteoglycan molecule called agrin. So these are specifically placed in the basal lamina where muscle and nerve are interacting. And this structure basically forms the function of a memory, allowing the cells to, when the cells have to restore the function, these molecules basically guide cells what to do. Let's look at that. The basal lamina separates the nerve and muscle cell, and this also, as I said, provides help to these cells to regenerate when there is a damage. So let's look at the damage. So we have a muscle and the nerve innervating that muscle. So if we get rid of the cell, the muscle cell is removed or destroyed, and so is the nerve is also destroyed. And it basically results in degeneration of muscle and nerve. The basal lamina can control the localization of acetylcholine receptors. Here, for example, in the blue, you can see those blue dots basically represent the acetylcholine receptors, which cluster in the muscle plasma membrane at neuromuscular junction. So both muscle and the nerve have been destroyed. Now we are going to allow only one of them to regenerate and see what happens. So if we allow the nerve to regenerate, the nerve will form the synapses at the old site, although there is no muscle there. This basically suggests that whatever was left in the extracellular matrix, which is in this case is the basal lamina, provided guidance to the nerve cell where it needs to form those synaptic structures. Let's do the reverse experiment. We are going to allow the muscle cell to regenerate, but we are not going to allow the nerve cell to regenerate. In this case, the muscle cells, when it regenerates, it forms, it clusters the acetylcholine receptors at the original site of the neuromuscular junction, although there is no nerve. So nerve is not giving the signals to the cell where to put these receptors. It is basically ECM, in this case the basal lamina. It is signaling cells where to put these specific receptors. So there is a lot more to basal lamina and extracellular matrix molecules than just the structural scaffolds. They provide signals. They can also remember the architecture. They can use this architecture when the cells or the tissue needs to regenerate itself.